My name is Professor Mate HT, Department of Botany, RBNB College, Sri Rampur. So today we are going to see the mechanism of opening and closing of the stomata, that is the stomatal movement. Because it is called as the stomatal movement because opening and closing of the stomatal aperture takes place. So there are the two movements, that is opening of the stomata and then there is a closing of the stomata. So first opening of the stomata. So this is the structure of the stomata. In the chloroplast of the stomata, already there is the starch. But when light falls on the stomata, that is on the guard cell, this starch is get converted into the glucose. It takes place in presence of the enzyme of phosphorylase and in presence of the light. And there is a formation of the glucose that takes place. And when glucose is get formed, it is when it is get synthesized in the guard cell, automatically the OP, that is the osmotic pressure of the guard cell, is get increased, and there is the water in the neighboring mesophyll cell or the subsidiary cell, and water from the subsidiary cell enters into the guard cell endoosmotically, and when water enters into the guard cell, then turgidity is get increased, and when turgidity is get increased, automatically turgor pressure is get increased, and because of this outer wall is get stretched toward the outer side. At the same time, the inner thick non-elastic wall becomes concave. This also happens in the neighboring guard cell, and the stomatal pore is get opened, and water loss in the form of water vapor takes place through the opened stomatal aperture. And in this case, when glucose is get synthesized, at that time the pH of the guard cell is get increased, and it is nearly about the 7, and it is also in favor of opening of the stomata. So it is only because of the synthesis of the glucose, and it is in presence of light. Because in presence of light, starch is get converted into the glucose, and OP is get increased, and as OP is get increased, endoosmosis takes place. That means the water from neighboring cell enters into the guard cell, and the turgor pressure is get increased, and then stomatal pore it get opened, and water loss, that is the transpiration in the form of water vapor, takes place. So in this way, stomata are get opened when there is light. Light acts as a sensor. Only in presence of light, stomata are get opened. So this is about opening of the stomata. Now the closing of the stomata. during night, that means when there is no light. In this situation, the glucose is get converted into the starch. Glucose is osmotically active while starch is osmotically inactive, and during night, that means when there is no light, in this condition, glucose is get converted with the help of enzyme phosphorylase and in absence of light into the starch. As the starch is osmotically inactive, it is not dissolved in the water, and because of this, the water which is already during daytime in the guard cell goes back into the neighboring subsidiary cell. That means exoosmosis takes place. Water moves away from guard cell into the subsidiary cell, and then flaccidity of the subsidiary cell takes place. And at the same time, when starch gets synthesized, pH also becomes acidic, that means it becomes a 5, and it is in favor of the closing of the stomata. And when starch gets synthesized, which is osmotically inactive, automatically exoosmosis takes place and flaccidity of the stomata takes place, and stomatal pore is get closed, and there stops the transpiration. This happens when there is no sunlight, that means during night. So this is about the closing of the stomata. Stewart proposed a hypothesis for opening and closing of the stomata in 1964. According to Stewart, in the chloroplast there is the starch, and this starch is phosphorylated with the help of enzyme phosphorylase into the glucose 1-phosphate. It is according to the Stewart's hypothesis. Already starch is present in the chloroplast of the guard cell, and this starch is phosphorylated with the help of enzyme phosphorylase into the glucose 1-phosphate. Now this glucose 1-phosphate is get converted into the glucose 6-phosphate. Here glucose 6-phosphate is there. It takes place because of the enzyme phosphoglucomutase. Now this phosphoglucomutase is dephosphorylated with the help of enzyme of phosphatase, and there is a formation of the synthesis of the glucose that takes place. Now there are the two substances, that is the starch which is osmotically inactive, and from this glucose is get synthesized which is osmotically active. That means when this glucose is get synthesized, the osmotic pressure of the guard cell is get increased, and as the osmotic pressure of the guard cell is get increased, the water from a neighboring mesophyll cell enters into the guard cell. Then turgor pressure is get increased, and when turgor pressure is get increased, automatically a pressure is exerted on the outer wall of the guard cell, which is thin and elastic, and it is get stretched toward the outer side, and at the same time the inner non-elastic thick wall becomes a concave. This happens also in a neighboring guard cell, and automatically the stomata are get opened, and the transpiration, that is the water loss in the form of water vapor, takes place. And in this case, that means when glucose gets synthesized, the pH also becomes 7, and because of this is also one factor which is responsible for opening of the stomata. Now this happens when there is a light. This happens when there is a light. All these changes takes place when there is a sunlight. But when there is no sunlight, at the time of evening, that means at the time of darkness, this glucose is get phosphorylated into glucose 1-phosphate with the help of ATP into the ADP, and the enzyme is hexokinase, and synthesis of glucose 1-phosphate takes place. Again there is the same enzyme is there, that is the phosphorylase. So this glucose 1-phosphate is get converted into the starch with the help of the same enzyme, that is the phosphorylase, and at this case the pH becomes 5, and automatically the exoosmosis takes place. That means the water from guard cell goes back into the neighboring mesophyll cell, and the flaccidity of the guard cell takes place, which closes the stomata, and here transpiration stops. This happens when there is no light, that means when there is no sensor. During daytime, stomata are get opened because of the glucose, because glucose is osmotically active. And during night, as there is no sunlight, that means if there is no sensor, this glucose is phosphorylated into the glucose 1-phosphate with the help of enzyme hexokinase, and finally there is a synthesis of the starch with the help of same enzyme, that is the phosphorylase. And starch, as it is osmotically inactive, the exoosmosis takes place, that is water from guard cell enters into the neighboring mesophyll cell, and flaccidity of the guard cell takes place, and automatically the stomata get closed, and here transpiration stops. So this is about the opening and closing of the stomata according to Stewart in 1964. Thank you.